Thank you very much. I'd like to thank Professor Pierre Mohammed and Dr. McNamee for the very kind invitation to speak to you and meet you all. I'm a bioinformatician, not at all in the world of physicians. So for us, this is really an interesting and hopefully promising interaction. So I'd like to present how we can use the entire genome to further precision medicine. As Professor Pierre Mohammed just mentioned, there's been lots of focus on gene sequences. Why? Because we can measure causality very strongly on these sequences. But there is also a lot of signal outside of the genic regions. So as Professor Roden presented just before me, there's quite a lot of large initiatives nationally, internationally, to collect massive cohorts and to study diseases, either phenotypes at large or specific diseases. For example, the Cancer Moonshot, ICGC, TCGA are collaborations which are specialized in cancer research. Conversely, DDD, Deciphering Developmental Diseases, is specialized in understanding child diseases. In particular, I'd like to mention the Global Alliance for Genomics and Health, which you and Bernie is currently leading, which is trying to create a network, an exchange network between all these organizations, as well as private organizations, basically defining the standards to exchange genetic and genomic information between institutions, taking into account legal and ethical frameworks. So to summarize Professor Roden's presentations very crudely, how can we go from GWAS studies, so genome-wide association studies, to a drug target? In short, we collect a massive cohort of cases and controls. We genotype them using some cool new technology. We then apply our knowledge of molecular biology running through the many databases stored at EMBL, the institution I come from. And then hopefully, this will lead to some new insight, to some new knowledge that will lead to new therapies, some new drugs, and in short, improve healthcare for society at large. And this is the ambition of the Open Targets collaboration. This is a collaboration between EMBL, so my institution, Wellcome Sanger Institute, and 5 pharmaceutical companies, GSK, Biogen, excuse me, I need to think, Celgene, Takeda, and Sanofi. And in the spirit of Professor Buntra's talk, this is a pre-competitive collaboration. Everything that's happening between these 5 companies and ourselves is then redistributed freely to the public. And so the goal of Open Targets is to develop resources in genomics which are relevant to discovering effective drug targets. And so one of the key resources, which is collaborating with Open Targets, is the GWAS catalog. This is a collaboration between the NHGRI, so this is a component of NIH, if you prefer, and EMBL, to collect all published GWAS results into a single database. You can go to the GWAS catalog website, and then you type your disease of interest, for example, IBD. And there you can see SNPs which are associated to IBD, along with some information about the p-values and all other kind of information about the gene targets and so forth. Now, we've got all these GWAS results. They're being produced massively across the globe. We're collecting them all in one location. Is that the end of the story? Can we just sit down and let physicians get on with their work? Unfortunately, there's quite a lot of work ahead of us. And this is where EMBL stands. So EMBL is the European Molecular Biology Laboratory - European Bioinformatics Institute. It aims to be Europe's home for biological data services, research, and training. In these uncertain times, it behooves me to tell you that we are European, but not at all part of the European Union. And regardless of what may be happening in Westminster in the next few weeks, we're here to stay. We've thrived in the British genetics world, and we're not giving up on that anytime soon. In fact, EMBL includes European states such as Switzerland, Israel, Australia. We've got discussions with India. So this is a very inclusive vision of Europe. So we are essentially a library for a library of biological data for all life scientists around the world. And this data is openly available to everyone. We're part of a greater organization, EMBL, which is headed in Germany. But here, or in Cambridge, there's 650 staff from 66 nations, and we're also in collaboration with Elixir, which is a network to exchange biomedical data. So our mission, as I mentioned, was to do some research, but that's actually a fraction of our mission. It's mostly to deliver scientific services to the world, deliver databases, data collections, and so forth, to train scientists, engage with industry, and also to coordinate bioinformatics in Europe. So the M in EMBL does stand for molecular, so we're very much focused on molecular biology. Once again, we're not physicians. We do not hold any medical records within our databases. However, we are specialists in how to sequence and how to analyze genetic data. And so one of the key applications, which you are most concerned with, is to interpret human variation. There's obviously quite a lot of focus on disease susceptibility and drug sensitivity. So within Ensembl EBI, the Ensemble Project is the project that I participate in. It is many things to many people. It's a website, which you can go to. It's a comprehensive database of functional annotations for genomic sequences. So we create information. We provide this information from the data, which is deposited into the EBI archives. It's a bioinformatic analysis platform, and it's a collection of integrated tools. So to go very quickly, we've got our website, ensemble.org. If you want, you can go on it right now. And there you can see, we've got more than 100 species. They're annotated in many different ways. If you don't like the website, don't worry. It should be changing very shortly. And sometime in the next 2 to 3 years, we should be completely revamping the website with a modern look and feel. So it should look like Google Maps. It should be way more interactive and sprightly. We're responsible to produce the Ensemble Gencode gene annotation. This annotation is the reference for all these molecular biology projects, so it is quite key in biomedical research. We produce evolutionary information by comparing all these species that we store within Ensemble. We've got more than 100 vertebrate species, more than 20,000 bacterial species, so there's quite a lot of evolutionary information stuck in there. More relevant maybe to this audience is a comprehensive resource to understand variation. So we could be looking at population structure, but also phenotypes, a bit on animal phenotypes if you're looking for a model, as well as published results. So typically, if you have a list of variants, for example, a 23andMe genotype, you could enter it in our most popular tool, the Variant Effect Predictor, VEP for short, which analyzes all the information that we have across our databases in the light of the variants which are entered. So for example, this variant is a coding mutation, this variant is on a regulatory region, and so forth. Now, how do we go from all this information to an actual phenotype? We can give you annotational information, but we can't necessarily lead you all the way to the result of these genotypes. Now, going back to this GWAS database, if you scour it and pull out all significant SNPs, you'll find that only a minority of them are coding SNPs, so only a minority of them can be interpreted quite reliably. The vast majority are intronic and intergenic. Presumably, they affect phenotype by regulating the expression level of nearby genes, or maybe the splicing of nearby genes. And there, the story gets a bit more complicated. So there's this famous story of the FTO locus that was published in 2011. There's a very strong association to obesity within an intron of the FTO gene, and quite reasonably, the authors of the publication said, well, it would seem that FTO has something to do with obesity. Now, a few years later at MIT, Melissa Klausnitzer and Manolis Kellis determined that the SNPs here are actually regulating the expression of RX3, this little gene here, and RX5, which is outside of this Manhattan plot. And so the story gets a little more shaded. It could be the expression levels of these small genes, which has an effect on obesity. Now, how do we interpret these non-coding variants? How do we determine what gene is being regulated by which SNP? And this is where my team comes in. So we've got to pull as much experimental data as possible, for example, epigenomic data, eQTL results, chromatin conformation data, as well, more recently, CRISPR and genome editing experiments. So the Ensemble regulatory build pulls in epigenomic experiments from the International Human Epigenome Consortium, IHEC for short. This is actually an umbrella organization that includes local consortia such as Encode, Blueprint, Deep in Germany, and so forth. So all these epigenomic consortia across the world are pooling their data within IHEC. We're managing actually the IHEC's metadata server, EPR. And so from this metadata server, we can then pull all the IHEC data and reduce it to a very simple annotation of the human genome. Roughly 20% of the genome, by our estimate, can be annotated as being, say, a promoter, a distal enhancer, a proximal enhancer, a CTCF insulator, and so forth. We try to determine the activity levels of these regulatory elements, a bit like genes can be expressed highly or lowly. These regulatory elements could be active, poised, repressed, inactive. And finally, there's an automatic pipeline, so we can just slurp in all the evidence, experimental evidence from IHEC, and it is introduced into this new annotation, which you can find here alongside the genes in the Ensemble browser. Another really exciting line of evidence are EQTLs. So to cut a long story short, an EQTL is an expression quantitative trait locus. This is, in other words, a GWAS for gene expression levels. So in particular, GTEx was a consortium in the US which collected samples from 1,000 donors across 44 tissues. And they measured their genotype as well as RNA-seq expression levels. Now, obviously, to get fresh RNA-seq samples, this was quite an impressive endeavor where you needed to obtain 1,000 recently deceased donors. And to give you an idea, they were at the entrance of an emergency room, and if somebody was recently deceased, they had 48 hours exactly to inform the family, consent the family, harvest the samples, and then perform the RNA-seq experiments. It's an incredible endeavor. It's an incredible data set. But that means that now across 44 tissues, we can say that, for example, the expression level of gene BRCA2 here is correlated to these SNPs or to those SNPs depending on the tissue of interest. So this is a fantastic data set to attach genotype to at least molecular phenotype. Another line of investigation, which is much more cost-effective, but slightly less reliable, more experimental, are chromatin confirmation assays. There's a few flavors of those: 3C, 4C, high C, chia pet. I'm probably missing a few. And in short, what they're doing is measuring physical proximity between certain pieces of chromatin. So basically, is your SNP of interest in physical proximity to the promoter of the gene that it may be regulating? There's absolutely no proof that it's regulating that gene. However, there is circumstantial evidence that it's loitering more than you would expect near the promoter, and therefore that would help you prioritize that variant. And so we've been collaborating with the Babraham Institute to test a new technique, promoter capture high C, which interestingly correlates really strongly with the epigenomic data. Finally, there's much ado in the news about CRISPR and genome editing experiments. Obviously, we don't really know where that world is going to go. But in the meantime, what we're doing is to collect all these experiments, a bit like the GWAS catalog. We have now the genome editing catalog, where our curator is reading all these papers and entering the key data into a single database, which will be available by the end of this year. And so we have all this data, both of associations, but also molecular data. How do we wish to analyze it? In short, we've developed, in collaboration with Open Targets, whom I mentioned before, an entire pipeline. So this is meant to be a turnkey pipeline. You could install it on your laptop, and pretty soon you can run it straight off the Ensemble web browser, where either you start from public data sets, such as the GWAS catalog, which I mentioned, or you might enter your own summary statistics. You've performed your own GWAS, and you'd like to know what's the causal gene. We perform an ontology search using the Ontology Lookup service. So this allows us to compare across databases that may be using slightly different denominations for the same phenotype. In some databases, a disease IBD might be abbreviated. In another database, it might be inflammatory bowel disease. So ontologies allow us to map these synonyms. We perform linkage disequilibrium expansion, so basically doing a genetics analysis using the 1,000 genomes data sets, which are stored at IGSR. Then we get basically clusters of SNPs, or GWAS peaks if you prefer. And there we throw the kitchen sink at them. We pull out all the epigenetic data we have, the Hi-C data we have, the EQTLs, the CHIAPA, external databases too, such as RegulomeDB, the variant effect predictor, et cetera, et cetera. And we integrate all this into a variant-to-gene interaction score. And then amalgamate it across your GWAS, you'll have an overall disease-to-gene association. So what we're saying is we don't have proof that the gene is causal of the phenotype. That'd be too good. But we do have strong evidence that the SNPs which are regulating that gene are also highly associated to the disease. So it's not quite a smoking gun, but there's very strong circumstantial evidence, which hopefully will be a strong lead for downstream research. And this led to the Open Targets genetics platform. I'm remiss, I forgot to put their URL, but it's targetvalidation.org. And so in this web browser, it is basically an amalgamation of Ensembl EBI resources, along with also Sanger data sets, which associate variants to diseases, or targets, targets to diseases. And they've developed, in particular, a genetics portal, so that is genetics.targetvalidation.org, where you can explore this data. So there's a very complex data set where you have to, your mind basically has to span between your targets, your genes, their regulatory variants, the GWAS variants, and the phenotypes. Now, thanks to great creativity and usage testing and really, snappy web design, we now have a highly interactive browser which allows you to explore this complex multidimensional data set. So in conclusion, Ensemble is aggregating numerous genomic data sets that will aid variant and GWAS interpretation, and we're bringing them together in tightly integrated services and resources, such as the variant effect predictor, which is already out and extremely popular, the post-GWAS analysis platform, which is in development, and the Open Targets genetics platform, which has just been released a few months ago. So with this, I'll thank my numerous collaborators. Thank you for your time, and take any questions you may have.